Hello everyone, and welcome to the Climate Change in America's National Park webinar series, where we invite guest speakers to join us every second Thursday of the month to share with us in the ways in which they're tackling climate change issues all over the nation and international parks. I'm Matt Hawley with the Climate Change Response Program here in Fort Collins, Colorado, and I'm really happy to have all of you with us today. I'm pretty excited about this topic that we've got. But before I launch into what we're going to talk about and introduce our speaker, I just wanted to give you a couple moments to let you know what this webinar series is all about, because I know at least a number of you are joining us for the first time. Basically, the way this works is that every month we do have a guest speaker or speakers to present an issue related to some aspect of climate change for up to about an hour. And then we open it up for questions afterwards and kind of a discussion period, giving a total of like a 90-minute chunk of time. We don't always use the full 90 minutes. I'd say most of the time we don't, but that's how much we plan on kind of blocking off the calendar. We do encourage you to ask questions and to make comments throughout the presentation. All of you attendees are muted by default. I won't be able to hear any of you, but you'll notice that questions box there. Right there on the side, kind of there on the tab, you have a questions tab, and that's your way of asking questions to us. I will receive all those questions and comments, and then I will relay them to our speaker. The way that we find it works best is you bring in the questions, I kind of keep them on hold, and I'll wait till the end when we're in that discussion period to ask them. Otherwise, it's just a little too complex back and forth trying to, oh, so-and-so typed this, oh, what do they mean? Nah, we'll just save them for the end. But keep them coming all the way throughout. We are recording this webinar today, and we do archive all of the recordings on our SharePoint site. You'll each receive a follow-up email after this webinar containing that link, so you can download the webinar video. Dr. Mazur's also been great. She's allowing us to download her PowerPoint presentation as well. So you will be able afterwards to reference back this presentation to go along with the video. So I want to tell you a little bit about Dr. Susan Mazur. She's been a professor of plant ecology, genetics, and evolution at University of California, Santa Barbara since 1988. Her research aims to detect the processes and results of evolution by natural selection, particularly for plant reproductive traits that contribute to the adaptation of plants to stressful environments, such as gas exchange rates, water use efficiency, speed size, age of flowering, flower size, pollen and ovule production. She's worked with a wide variety of species and plant communities to detect reproductive and physiological adaptations, ranging from South American tropical rainforests to the Sierra Nevada and coastal ranges of California. Her current research is investigating the factors that influence the evolution of mating system, physiological performance, age at flowering, phenology, and drought avoidance in the California native wildflower genus Clarkia. So we've got someone who's incredibly well-rounded and knows her subject very well. And I'm going to throw the ball over. Susan, are you there?
I am here.
All right. I'm going to make you presenter, and you can take it away.
Okay, let me know when you can see my first slide. Oh, it looks like I have that power.
Okay, we see your desktop now, so go ahead and maximize that, and we'll be ready to go.
First, oh, there we go. Okay, good morning, everyone, and thanks so much for taking the time today to learn a bit about the California Phenology Project. I'm going to refer to the California Phenology Project throughout this talk as the CPP, and that will enable me to shave about 45 seconds off the presentation. Thank you especially, Matt, for arranging the seminar. And I'm going to try to achieve 3 goals in the next hour or so. First, I'll describe the background, the objectives, and the scope of the CPP, which, as you'll see, was designed or was created to design and to test a protocol for phenological monitoring and education programs in 7 pilot parks. But these are programs that we hope can be applied to any national park public land, private reserve, schoolyard, or residential property. And I'll mention as we go some examples where this is proved to be true. Second, I'll present very briefly, and I hope with a minimum of confusion, a few of the somewhat complex analyses that I've conducted so far with the phenological data that have been contributed by participants in the CPP. And these analyses are designed to detect the relationship between local climatic conditions and the timing of phenological events as a first step towards predicting the effects of future climatic conditions on the phenology of our targeted species. And third, I'd like to hear your thoughts regarding, once you've heard this, regarding the potential to begin or to continue phenological monitoring at your site or with your organization if you're associated with an agency or institution other than the Park Service, and to brainstorm a bit about how to overcome potential obstacles for doing so. Sometimes people are so overwhelmed by the scope of the project that they feel like it would just be impossible to take the steps to start it from scratch at their institution. And over the 5 years so far of this project, we've discovered a number of ways where it's possible to simplify the protocols and scope of the project at any particular location to make it highly manageable to begin to track the effects of climate change on even a small portion of a park's flora. So don't be intimidated if this seems overwhelming. Just let us know at the end and we can start brainstorming about how we could modify the CPP's activities at your location. So the title, as you can see, is a rather long one. And the first 90% of the presentation is really aimed towards describing the first 4 words in this title, that is the scope and content of the California Phenology Project. And it'll only be towards the end that I will spend the time to share with you some of the species-specific phenological patterns that we've seen linking climatic variation to phenology in 7 of the national parks in California. Okay, having trouble forwarding this. Hold on a sec. Spinning beach ball already. There we go. Okay, so the California Phenology Project, or the CPP, was designed and conceived as a statewide phenological monitoring program to track the effects of local and recent climatic conditions, as well as in the long-term climate change on the seasonal cycles of wild plant species. My colleagues in the participating pilot national parks and I were very fortunate and grateful to receive funding from the Climate Change Response Program, and that effort really was spearheaded by Angie Evenden, the research coordinator of the California Ecosystem Studies Unit, and she was really a wonderful and visionary leader of this project that helped everyone contribute, I feel, in the best way possible. So we had several goals that we stated at the outset. Our scientific goals included establishing phenological monitoring and education programs in 7 pilot national parks, and this was soon extended into the University of California Natural Reserve System, a couple of botanical gardens, a number of schools, where we used the same standardized protocols that were created and which we helped to customize for California, created by the National Phenology Network, which is sort of the mothership that has been guiding and supporting us throughout this whole process. We wanted to cover a large geographic area and sample species and individuals across broad environmental gradients so that we could capture a good deal of climatic variation with respect to both rainfall and seasonal temperatures. And of course, the overarching goal was to assess the effects in a quantitative and rigorous way, the effects of local climatic conditions on the timing of seasonal events such as bud break, flowering time, fruiting, seed dispersal, etc., in order to predict responses to climate change. Of course, being a publicly funded project, we also had a number of public service goals, including to inform resource management decisions as appropriate at each park, to educate park visitors from organized school groups to individual hikers and drive-through visitors. We wanted to help prepare the public and park interpreters to observe and to describe changes in the landscape so that they would recognize that the park's floristic resources are not just a green backdrop to the more interesting animals that may walk in your path once in a while. And in some ways, most importantly, we wanted to engage citizen scientists in genuine research that would contribute data that would be used in ongoing analyses. No surprise, we have a website. What you're looking at here is the website that we've been using for the last 4 years that has been updated, but the update has not replaced this website, and I'll show you the differences very briefly right now, and then we can explore them a little bit more in the discussion period. So take note of the URL shown here, and just take note of the individual tabs. In the Meet the Species tab, you can gain access to each of the 30 species that we've been monitoring in the parks for the last nearly 5 years now. In the Phenology in the Parks tab, you can be introduced to what each of our 7 pilot parks has been doing and the species that they've been monitoring. In this tab as well, you can find both Google Maps and PDFs of static maps that users can print out to find each of the individual labeled plants that we're monitoring. In the Education tab, there are a wide range of lesson plans, seminars, and a variety of other materials that are appropriate from the K through 5 level through the university level for phenology-related instruction and activities. The Participate tab leads you through the steps, most of which are on the website of the USA National Network, the steps towards participation. This now is, what you're looking at now is the updated California Phenology Project website, which is similar to the website that I just guided you through, except there's now also a partners tab, and I'll illustrate that for you towards the end of the presentation. And here too you can see the Phenology in the Parks tab, which is where you would be introduced to our activities in each of the 7 parks. In the Partners tab, we now have 11 participating partners, which comprise some national agencies such as the Don Edwards National Wildlife Refuge in the Bay Area, a number of based educational programs such as Nature Bridge in Golden Gate and the Santa Monica Mountains, several of the UC reserves, and the Santa Barbara Botanic Garden. In the resources page, which has been updated quite a bit, I would urge you to explore that to find, among other things, our recently created project briefs, links to each of our peer-reviewed publications created during the term of the funding period, some informal publications and internal publications internal to the National Park Service that we created that provide information about how we selected species, best practices, our scientific framework, and a couple of progress reports. And then we have PDFs of site-specific project reports created by interns and staff members at several of the contributing parks. Okay, so some vital statistics in a nutshell. Throughout the duration of the project, we've been monitoring a total of 30 woody species, shrubs and trees, in 7 national parks in California. And we chose woody species to focus on for the last few years because we wanted to map and tag in a semi-permanent way individuals that we could monitor year after year, so that should we find that they exhibited phenological changes, that is, changes in the annual onset date of different phenological phases, we would know that that change was environmentally induced rather than due to a change in the specific biology of or changes in the individuals that we were monitoring. Now, of those 30 species, 8 of them are monitored in multiple parks, thereby providing the greatest geographic variation that we were able to obtain. We mapped and tagged over 950 individual plants in those 30 species. And to date, the participants in the California Phenology Project, which include primarily visitors and staff members in the national parks, in the 7 national parks, but also a good number of people in those partners that I introduced earlier. We have contributed over 1,200,000 observations to the National Phenology Network database. I'm really happy and proud of this effort. The California Phenology Project effort accounts for about 20% of all observations nationwide that have been contributed to the USA-NPN database over the last 5 years. These are observations apart from, say, legacy data sets that have been contributed by the Clonal Lilac Project, for example, but they represent on-the-ground observations by people who have been trained by members of my lab and by staff members in each of the participating national parks. After the first 3 years of data contribution, we started collecting, hello, oh, sorry about that. After 3 years of data collection, we started analyzing the data, some of which I'll present today, and found that we could indeed start to detect highly statistically significant associations between phenological onset dates, that's the day of the year on which a phenophase was first appeared, and climatic variables such as seasonal rainfall and monthly minimum temperatures, just as one example. Okay, so we monitored, or the 7 pilot parks in which we established our monitoring and education programs are shown here, going from north to south. Redwood, Lassen, Golden Gate and its associate, the John Muir National Historic Monument, Sequoia and Kings Canyon, the Santa Monica Mountains, and Joshua Tree, giving us 2 montane parks, 3 coastal parks, 4 if you include John Muir, and then 1 desert park. And at each of these parks, we had some super important local heroes without which we never would have been able to achieve our goals. The primary contact and leader at each of the parks are shown here. Most of these people are staff members in they're vegetation ecologists or botanists in the resources division, although some of these people like Christy Brigham have now been promoted to more responsibility. But without each of these people promoting phenological monitoring, helping us to find interns and volunteers to dedicate their time to the project and to serve in many organizational capacities, including hosting the 60 or 65 full-day workshops that we offered in these parks over the course of the 4 years of the project. Without them, we wouldn't have been able to get very far. Without the countless numbers of seasonal interns, contractors, and volunteers, the data just would never have been collected. So I really had my eyes open to how much work goes into implementing any kind of program in the national parks, particularly a program like the CPP, which was not a top-down mandate, but which depended existentially on the goodwill, extra time carved out of their already overwhelming schedules, and volunteer efforts of the people and groups named here. About halfway through the 5-year period, we also started training people at a subset of the what are now 39 University of California natural reserves. And those shown here, which were more or less accessible from my research group's location in Santa Barbara, we trained volunteers and local staff members to contribute phenological data on the same species that we were monitoring in the parks, thereby extending our reach and the depth of our data collection. Okay, so I'm going to run very briefly through the primary steps that we engaged in the last 5 years. And in each of these, for each of these elements, I'm really only going to be able to skim the surface, but you'll get a flavor of each of these activities and where to get more information. So our starting point was to identify scientific questions that we felt would be tractable within the limited term of our funding. Examples of these questions, which came out of a one-day meeting that we led at Berkeley, that included University of California faculty, agency botanists, and national park colleagues early in the project in November 2010, I believe. We articulated a wide range of scientific questions that we felt that we could address and prioritize, a very few of which are shown here. So first and foremost, we wanted to ask how iconic and widespread species in California respond phenologically to environmental variation and to climate change, even within the 4-year duration of the project. We wanted to design a program in which we could ask which taxa or functional groups, say shrubs versus trees, evergreens versus deciduous taxa, semi-deciduous versus fully deciduous, how these different functional groups are responding to climate change or might respond to climate change. With a geographic range that we had represented among our pilot parks, we felt we could address whether communities or plant communities or distinct habitats differ in their responses to climate change. If we got really lucky, we thought we might be able to begin to address whether relationships between plant and animal mutualists, such as plants and their pollinators, are disrupted by climate change. And when I introduce you or help you navigate the new website, I'll show you where the document that we created describing our scientific framework, where that can be found on our website. If you ever have trouble sleeping, that's a really good way to cure that. Okay, so how did we select our focal species? This was a several-month effort that started with the perspective that among the species selected for monitoring the CPP, we wanted to include a variety of attributes. And we knew that no single species would be able to fulfill all of our priorities for selection criteria, but collectively, we wanted our focal species to be able to be represented, to represent a number of priorities. So, for example, we wanted to choose among our species at least some species that were locally dominant or that were good indicators of a particular habitat type. We wanted a good number of our species to be widely distributed so that we could capture a good deal of geographic, environmental, and climatic variation. We wanted to include some species that were of management concern. We did feel very strongly that all of the species had to be easy to identify and had to have a strong ability to engage citizen scientists. Species that we knew that species that would be frustrating to identify, we might be able to get people familiar enough with them at a particular site to monitor them carefully, but that such species were unlikely to be monitored more broadly if they were difficult to identify unambiguously. In some cases, we aimed to choose sites and species that were close to other monitoring efforts, or such as air quality, as well as basic meteorological data. We hoped to include some species for which there are legacy data, such as fantastic herbarium collections. We thought we might be able to choose some species that were benchmark species, that is, those that very closely track the transition from winter to spring and would be good examples of first responders to spring warming. But we also wanted to include some species that flower very late in the season, so that we might understand how drought would affect species that flower, say, during mid or late summer. And then, of course, we also had to make sure that in the parks in particular, that the species and individuals that we chose were in known and in accessible locations. We learned very early on in our discussions with botanists at each of the parks that the last thing we wanted to be responsible for creating were a series of new social trails that would promote erosion and wandering by the public. And so it became really evident to us as we pondered all of these priorities that really the two most important criteria to fulfill were ease of identification and the distribution of these plants in known and accessible locations, and then we could work on fulfilling the other criteria as well. So fundamentally, after we meaning myself, Angie, the vegetation ecologists and botanists and plant-friendly educators in each of the national parks that we visited, and then a wide variety of botanists in federal agencies and universities across the state who we surveyed in through a fleet of email messages and Excel spreadsheets. We managed to whittle down the over 5,000 native taxa in the California flora to 75 species, any of which would have been totally swell to monitor. But among those 75, once we visited each of the parks and considered their accessibility and ease of identification, those were further filtered to our final targeted list of 30 species that we're currently monitoring in these parks. And so as you can imagine, there were many trade-offs that we had to make among the criteria that I listed before in order to choose species that represented a compromise among all of our goals. And of course, among these 30 species, maybe the best sort of litmus test of our success is that nobody was happy with all of them. And that's just sort of par for the course when trying to engage many people, each of whom have our own favorite genus or habitat, to agree on a very, very short list of species among such a phenomenal candidate list. Okay, so as I mentioned earlier, you can learn about these 30 targeted native species by going to the Meet the Species tab of the CPP website. Now I want to just for a moment put these 30 species in the context of all of the species that are being monitored nationally by the USA-National Phenology Network. So the USA-National Phenology Network, or NPN, has targeted over 650 plant species nationwide that anyone can contribute phenological data on using the wonderful, very user-friendly interface called Nature's Notebook. And this is the interface that we use in the CPP as well. Our data enter the NPN database in the same way that any contributor to the National Phenology Network contributes their data. Now, of these 650 plant species, more than 340 of them also occur in California. So California represents, or at least includes, a huge chunk of all of the species diversity that's being monitored nationwide. And the 30 species that we're monitoring in the CPP, therefore, are both a subset of that 650 species list and the 340 that are in California. Okay, so we had some easy choices and some hard choices. It was evident from the very beginning that we were going to monitor Joshua tree, Yucca brevifolia, in Joshua Tree National Park, right? Even though this species isn't found in any of our other pilot national parks, there was no way that we weren't going to monitor Joshua tree. It's iconic. It has a obvious ability to address scientific questions. It's a very sensitive species. It's distributed near other monitoring efforts, including demographic studies of Joshua tree itself. It's a great local management concern, and it can engage citizen scientists point blank. Everyone in California, if they know any species, they know Joshua tree. We thought at the very beginning that surely we would also monitor the common yellow monkeyflower. It's widespread, it's widely studied, we know a lot about its pollination biology, its mating system, but there are many other yellow monkeyflower lookalikes, so the possibility of incorrect identification, particularly off-site of the parks, was very high. And more importantly, it prefers wetland habitats, stream sides, seeps, temporary or permanent wetlands. And it was evident very early in our conversations with park managers that they and we did not want park visitors to be marching heavily into sensitive wetland habitats. And so we nixed this from our list of favorites pretty early on. We also chose to monitor creosote bush, not because it's the most dramatic and charismatic species, although once you look closely at its flowers and really adorable fruits, everyone gains quite a bit of affection for it, but also the widespread nature of the species and its value as an indicator made it one that we could all agree on very early on. Blue oak, iconic species of the foothills, present in a number of parks and a good number of our partner sites. We knew right away that this would be a good candidate for monitoring. Eriogonum fasciculatum, also widespread and with a number of easy to identify phenophases, was also a clear candidate early on. Blue elderberry as well, easy to monitor. Its phenophases are quite unambiguous, was another plant that went on our list early and stayed there with little argument from anyone. And I'll talk about some of those species later on when we get to the data analyses. Oh, and then finally, coyote brush, Bacchus tiliolaris, was a favorite species for a number of reasons. First, it's very, very widespread. Second, it flowers in the late summer, early fall, therefore being one of our late season species. And then finally, it's a dioecious species, meaning that individual plants are either male, here you see the anthers emerging from the individual flowers. Here are the female flowers. Plants are either male or female, and we thought this might give an opportunity to detect gender-specific responses to climate change, which would be a novel and intriguing possibility. Okay, so just a few words on why you might want to monitor one of our 30 focal species instead of one of the other 300 some odd National Phenology Network species in California. First, we created over the course of the project a number of species-specific monitoring tools, including 2-page documents that we call species profiles. I'll show you an example in a moment, that are available to download from the CPP website. And these greatly facilitate the standardized, consistent monitoring and detection of different phenophases in each of our species. They're basically photographic guides to the phenological phases. Also, if you choose one of these species, you will know that if you observe them at a new site, at a new park or at a schoolyard or a private reserve, that those data, the data that you might contribute, complement CPP data that have already been collected at national parks and University of California reserves, collectively contributing to our understanding of how these taxa respond to environmental and climatic variation. And also you can benefit from the collective experiences of the California Phenology Project Observer Network. And there's a listserv that is monitored and administered by staff members at the National Phenology Network that helps phenology leaders, or really any volunteer, to benefit from the experience of others. And there's a link to how to join that listserv on the CPP website. And I would urge you to do so if you're interested in partnering with other people and learning from their experiences. Very early on in our work, it became evident that we needed to work with the USA-National Phenology Network to develop some phenophase descriptions that were appropriate and, in fact, necessary for California plant taxa. If you've monitored species or just been an observer of the seasonal cycles of plants in the temperate zone, in the northern Midwest or on the eastern seaboard, you will generally expect in the wintertime to see woody plants produce large buds, which in the spring synchronously open to reveal developing leaves or opening flowers. Well, we discovered that in all but 2 of our 30 focal species, the plants do not produce large buds in the wintertime or in the fall to overwinter as buds which open in the spring. Rather, because few of them are limited by frost and thereby need to produce these buds, the production of young leaves is a process that occurs year-round in response to recent rainfall. And as a result, we needed to learn to help people recognize the appearance of young leaves using definitions that don't apply to large bud-producing species in the temperate zone. And it took quite a while for us to wrap our heads around why some of the phenophase descriptions that have been created for New England and the Midwest weren't working for our woody species in California. But with a lot of back and forth and input from our park colleagues and from park visitors and interns, we worked closely with the National Phenology Network to tweak and to revise the phenophase descriptions for the applied to the peculiarities of the California species and really to all species in Mediterranean systems. And I'm really grateful to the members of the NPN, particularly Ellen Denny, Kathy Durst, Jake Weltine, Teresa Crimmins, for supporting those revisions that that we had to create in order to use the protocols of the NPN as appropriate for California species. We had hoped from the beginning that we might be able to identify and use historical data sets to take advantage of the fact that photographs and herbarium specimens captured phenological information in a way that we could take advantage of. And I don't have time to talk very much about this, but while we didn't have the time to discover photographic records that we could use, as Abe Miller-Russian has done, to demonstrate wonderful examples of changes in phenological patterns in the Boston area. We do in California have an extremely well-organized electronic database representing the millions, literally millions, of herbarium specimens that are present in our wonderful herbaria. Liz Matthews, who was my wonderful companion and postdoc in this project, she and I dedicated quite a bit of time to training undergraduates at UCSB who monitored the phenological status of over 4,000 specimens in 4 species, not woody species, but herbaceous annual species or short-lived perennial species, where the herbarium specimens display very clear phenological information along with the data collection. We are still in the process of analyzing data for Lupinus, Clarkia unguiculata, and Clarkia cylindrica, but we've published one paper on the phenology of Trillium ovatum, a actually not so short-lived perennial plant that has the very convenient attribute that individual stems typically produce only a single flower, thereby allowing us to detect, based on the specimen label data, the date on which flowers were fresh going back 150 years. And we recently published this, actually just in the last month, a paper in the New Phytologist in which we demonstrated that over the last 150 years, changes in the timing of flowering in Trillium ovatum is a function of strong interactions between local temperature and local precipitation in western North America. And here you can see the distribution of the herbarium specimens that we sampled for this project. So stay tuned for more work on this as we make our way through the other data sets. Okay, so now we get to our work in the parks, where not only, as I mentioned earlier, did we have to develop and revise the monitoring protocols initially created by the National Phenology Network, but we had to test them with park staff and visitors to see if we needed to tweak them in some way that would facilitate their use. And I'll walk you through some of our activities along these lines. So what you're looking at here is a 2-page species profile for coyote brush, which is an example of the species profiles that we created and that are available online on the CPP website for each of the 30 species that we monitored in the CPP. So the first page has an introduction to the species. The second page has photographs and generally helpful hints, usually helpful hints, for how to unambiguously identify each phenophase whose appearance is targeted for recording in the National Phenology Network's database through Nature's Notebook. And so typically new users or new participants print out these guides to take in the field with them the first few times they monitor a given species, and then eventually they can be, they don't need these anymore. Now, all of the species monitored by the NPN are monitored in the same way. This image displays a data sheet, which in this case is specific to coyote brush. Now, while the format of every data sheet is identical, depending on the life form or functional group of a given species, the particular combination of phenological phases that are listed here differ somewhat. So, for example, coyote brush does not produce or produces quite copious pollen, and as a result, we do monitor the appearance of pollen release on this species. In other broadleaf evergreen species that do not produce copious pollen, pollen is not released. So the National Phenology Network has classified each of its 350 species in California, 650 species nationwide. Each species is placed within one of about 25 different functional groups that provides the list of phenophases that are to be monitored. Now, while this data sheet looks somewhat complex, what one is required to do on a given date of monitoring is simply to answer yes or no to a series of questions that begins like this. Do you see young leaves on that date? Yes or no? Or are you still unsure how to identify individual young leaves, in which case you would circle the question mark. Do you see flowers or flower buds? Do you see open flowers, etc.? Along with these data sheets come very detailed and consistent descriptions of each phenophase, which again are used by participants the first few times they monitor a particular species, but very soon they're weaned off of those descriptions and they can proceed into the field just with these data sheets. I would hasten to say also that there's now an app called Nature's Notebook that you can download from the App Store for both Androids and iPhones, in which the interface looks nearly identical to the paper data sheets, so that if you're actually, whether or not you're in a region with cell phone reception, you can enter data with your cell phone and it will be uploaded once you're back into cell phone range. So these data sheets are very easy to use once you've gained a little bit of practice and used them along with the species profiles and the phenophase descriptions. Well, when we visited each individual park to begin with, after Liz and I and at the first year of the project, Brian Haggerty, who's a PhD student in my lab, we consulted in depth with local botanists to identify trails and visitor centers where we'd be able to monitor between 3 and 7 of our focal species. We started out with hand-drawn maps and GPS coordinates of each of the individual plants that we located and selected for monitoring. And then back in my lab, we converted these into Google Maps as well as into static maps that I'll mention in a moment. This is Josh Hoynes observing a yucca in Joshua Tree National Park and possibly placing a identification tag on it. As we monitored individual plants, we tagged them with easily removable metal tags. We know that these are easy to remove because park visitors would occasionally remove them without permission, and they would occasionally have to be replaced. But each individual plant has a long code that gives detailed information about where it's located, as well as a unique 3-digit identification number. Along with the Google Maps, at the parks, we created these static maps at different scales. This is the map for Joshua Tree, which shows our 4 major monitoring locations. And then for each location, we created a large-scale map, such as this one, which shows each of several sites and the color-coded positions and codes for each of the individual species that are monitored at an individual site. And so these maps would, again, be printed out and used by participants as sort of a treasure hunt map for the plants that they were committed to monitoring. And eventually, within 2 or 3 weeks of monitoring, these maps were almost unnecessary by users because they would know the locations of all the maps of all the plants that they were assigned for monitoring. And this is just another example of another set of map locations along Park Boulevard at Joshua Tree. And another example in the Presidio. Oh, sorry, these are the static maps, and then this is an example of the Google Maps available online, and I'll illustrate that for you during our short tour of the website after the presentation. Okay, so in the final section of the presentation, I'm going to go fairly quickly through some analyses of our data to date, where I aim to detect relationships between local climatic conditions and the onset of phenological events. And this is an ongoing work in progress, but the basic goal of these analyses was to ask, and answer, how do local climatic conditions, namely recent monthly temperature and rainfall, conditions preceding leafing and flowering, affect the onset date, that is the day of the year from January 1st to December 31st, of targeted phenophases. Now, the basic quantitative scheme for addressing this question is to use a multiple regression approach. There are other ways to do it, but the examples that I'm going to show you here use this format, in which we ask whether we can predict the day of year of the onset of a given phenophase from a monthly parameter, such as minimum temperature or maximum temperature. And this equation is basically the equation for a line, where y is equal to the day of the year. The temperature parameter is the independent or the predictor variable, and this alpha value represents the slope of the line, and the b is the intercept. So we've got y equals mx plus b from algebra. Now, as you can imagine, the day of the year is likely to depend on more than one variable, more than, say, the monthly temperature, right? And so, for example, we can write a much more complicated equation where we ask how the day of the year depends perhaps on monthly conditions in January, temperature conditions in January, temperature in February, temperature in March, maybe rainfall in March, and ask how each of these parameters independently contributes to our ability to predict the day of the year on which a particular phenophase appears for the first time. And so for the analyses that I'm going to show you, I used day of the year of either leafing or flowering as the dependent variable, and I used a suite of independent or predictor variables, including minimum or in some cases maximum temperature for each of the months preceding the spring, and I also included precipitation. And then what I did was used stepwise regression to identify which of these variables explain a significant portion of the variation in the day of the year of the appearance of a given phenophase. And so in any particular model that was ultimately selected, only a portion of these variables might persist as being statistically significant. And I'll show you some examples now. If you're interested in reading about these in more detail, these were these analyses were published in June in a paper that I wrote with Kathy Gerst, Liz Matthews, and Angie Evenden. That was published in Ecosphere in June of this year. Okay, so starting with coyote brush. This was probably our best monitored species. We had sites that were monitored twice a week during its phenologically active periods, from Redwood to Golden Gate to the Rancho Marino, a natural reserve, part of the UC system, Coal Oil Point, also a UC reserve, and the Santa Monica Mountains. And when I ran the stepwise regression on to evaluate the variables that predicted the onset date of the appearance of young leaves across all of these sites, I found that only 2 of the variables were retained in the model. And what this analysis told us was that leaf production, the appearance of leaves in coyote brush, is highly rainfall dependent. Precipitation, particularly in March, was the best predictor of the onset of leaf production. And this positive regression coefficient indicates that sites that had the most rainfall in March had the most delayed appearance of young leaves. In other words, wet early spring at a given location resulted in late onset of leaf production. What about flowering? In this case, a different suite of predictor variables were retained in the model. And interestingly, the significant predictors in this case had more to do with temperature than rainfall. In other words, flowering time in this species was temperature driven. But curiously, the direction of the effect of warmer temperatures depended on the month. So what this regression coefficient tells us is that warm Decembers with higher average minimum temperatures, were associated with earlier flowering, while sites with warm January conditions were associated with later flowering. In other words, minimum temperature has opposite effects on flowering time in different months. And this was a surprise to us. We had no a priori expectation that at a given site, the conditions under different months might have highly different predictive effects on the onset of phenology. I would also point out that these significant regression coefficients down here reflect strong interactions between winter temperature and rainfall. In other words, in addition to being able to significantly predict the onset of these phenophases based on the average temperatures of December and January, there are strong interactions between temperature and rainfall that must be considered if you want an accurate prediction of the flowering time of this plant. Okay, taking a look at valley oak. Here, among our monitored sites, there's much lower geographic range. However, in the Santa Monica Mountains, we have quite a bit of elevation range among these sites, so we still did capture quite a bit of variation, climatic variation among the sites and individuals monitored. Here, when we looked at the model that best explained or predicted the onset of breaking leaf buds, 4 of our original monthly variables were retained in the model, including both temperature, spring temperature, and rainfall. And again, we see the situation where different months have opposing effects on the onset dates of breaking leaf buds. Wet Decembers and wet Marches delayed leaf bud production, while wet Februaries accelerated it, meaning predicted earlier bud break. But broadly speaking, the timing of bud burst is precipitation driven. Again, highly month-specific effects and important interactions significantly contribute to those onset dates. If your head is spinning at this point, you're in good company. I would hasten to say that very few studies to date have tried to detect and tease apart month-specific effects of rainfall and temperature on phenological onset dates. And as complex and confusing as these results are, we're also very gratified that even with just the 3 years of data that contributed to these analyses, we were able to detect such statistically significant results. I'm not going to show you the detailed results for Eriogonum fasciculatum or for blue elderberry, but I have just a couple of summary slides to end with. So when we looked at the outcome of all of these stepwise multivariate models for each of the up to 5 phenophases that we evaluated, we found that the ability of these models to explain the total variation in the onset dates of each of these phenophases was really quite high. So for example, 70% of the variation in the onset date of breaking leaf buds in blue elderberry could be predicted by those 8 variables that I mentioned, the 4 rainfall variables and the 4 monthly temperature variables. All of these R-squared values are incredibly high for multivariate model, well, for ecological data. And so I think that we can expect with the continued accumulation of data from the California Phenology Project, we'll be able to improve the models and get a better idea of the general climatic conditions, for example, that account for early versus late flowering and bud break in valley oak and in each of our widely distributed species. When I put together the data and the regression coefficients from all of the models, I was able to discern a few patterns that I found pretty interesting. Based on the geographic variation that we were able to detect in climate among the sites, in general, warmer winters, warmer monthly temperatures among the sites of coyote brush were associated with early flowering, while drier winters were also associated with early flowering. So what I think this means is that based on the data we have to date, we would predict that as future conditions get warmer and drier, we can expect that coyote brush will flower early. Well, not all of the species showed such consistent results. In Quercus lobata, warmer winters resulted in, or sites where the winters were warmer had later leafing out, later flowering, and later fruit drop, acorn drop, than cooler sites, and drier winters, sites with drier winters had later leaf out and later flowering. So this is a consistent result. We might expect in a warmer, drier future, while Baccharis flowers earlier, Quercus lobata flowers later. An example of an inconsistent result relative to what we expect in a hotter, drier future is shown by Eriogonum fasciculatum, where warmer winters are associated with later flowering and later fruiting. That is, the sites with warm winters flowered later than the sites with cooler winters. But the sites with drier winters flowered earlier and had earlier fruit ripening than wetter winters. which collectively means that in a warmer, drier future, we're not sure which will prevail, later flowering or early flowering. And then finally, for Sambucus nigra, warmer winters resulted, or sites with warmer winters are associated with earlier leaf out, but no effect on flowering, while sites with drier winters had earlier fruiting. So what does this mean in California in general? It looks to me, and of course this is a very tiny sample size in terms of the number of species, but if we were to make projections for what we might expect in a hotter, drier future, some species flower earlier, some species flower later, which at the community level, if I were to really go out on a limb, implies that we may see an overall changing in the timing of floral and vegetative resources, which could mean a compression of the duration of flowering. Baccharis is a late flowering species, and it would appear to be destined to flower early, while Quercus is a spring flowering species and may be destined to flower later, so that collectively there's actually a compression of flowering at the community level. That's a very tenuous projection, but it's the sort of prediction that we might be able to confirm with continued analysis of the data. So to summarize the analyses to date, we have been gratified to see that these multivariate models can account for a very high proportion of the variance in spring phenology. That doesn't necessarily mean that we'll be able to make predictions that are highly accurate, but we can begin to detect the climatic parameters that are important for different species. We've seen that associations between monthly temperature and precipitation conditions and the onset dates of subsequent phenological events differ among phenophases and species. And so although many studies across the US and in Britain have predicted generally that warmer winters will result in earlier flowering, and I'm sure you've heard of many of those examples, in California and perhaps in many Mediterranean climate locations, the relationship between warmer winters and flowering is not going to be so simple. We may see that if early flowering species flower later and later flowering species flower earlier, that we may see a thinning of floral resources at the beginning and end of the season, and we don't know quite what that might mean for pollinators and herbivores. But I hope with continued analysis of these data, I'll begin to be able to find these out. And I want to mention that Kathy Gerst and my graduate student Natalie Rossington and I are now beginning to analyze the two best monitored California oak species, Quercus agrifolia and Quercus lobata, along with two very well monitored species in the central and eastern portion of the US, Quercus rubra and Quercus alba, so that we may begin to be able to detect broader regional and continental differences among closely related species in their responses to climate variation and ultimately climate change. And finally, I just need to thank again all of our local heroes who helped organizationally and on the ground, with a special shout out to John Gross, who helped us obtain climatic data from the PRISM database, Catherine Thomas, who was highly instrumental in helping us to identify species for monitoring within and across the national parks, and Jake Beltine, the director of the USA-National Phenology Network, whose support and encouragement through this process has been really inspiring and helpful. So I think I'll end there. I do have my other sort of agenda item is to give you a tour of the new and improved website, but I'm of course most interested in your questions and responses as well. So I'll end here.
Great, thank you. Yeah, we've got about 15 to 20 minutes or so for questions. So thank you for that overview, Susan. We've actually had a number of questions coming in, and just want to remind people that you can just expand that questions box in your control panel, type them in, they'll come to me, and then I'll relay them for there. So I wanted to start basically talking about finding species in accessible locations as one of the variables you had talked about. And how did you ensure that like human-created openings and like roads, et cetera, were not affecting the local microclimates at those spots?
So this is always a potential issue, and of course we can't rule out any anthropogenic influence on plants that are accessible to humans. So once we had our list of 30 species, Liz and I went to each park and had brainstorming sessions with anywhere from 3 to a dozen botanically cognizant members of the Division of Resources, Education, and sometimes the interpretive staff as well, to discuss the selection of species and locations. At most of the parks, maybe even all of the parks, we knew we wanted to have at least one location, a location being a trail or a visitor center, across which there are multiple, local sites, a site being maybe 50 meters in diameter, or 50-meter length of trail where we wanted to monitor plants. So we always chose a visitor center because we knew that we'd have great access through the interpreters to small groups of visitors. And so those are places where, sure, there may be local heat island effects due to parking lots. There may be soil compaction effects that we felt, were unavoidable, and we had to accept them in the interest of being able to educate the largest number of people. So at all of the parks, the places where we monitor comprise a mix of fairly heavily impacted sites to sites that have sort of a minimum of trail traffic. This is a problem, or not a problem, it's a challenge for all of the plants monitored nationwide. They're all accessible to humans, so they're all vulnerable to some kind of human impact. Ultimately, because we monitored between 30 and 50 individual plants at each park, we have some reasonable sample so that the phenological window that we have on that park is not highly vulnerable to the damage that one particular plant might have incurred in its whole lifetime because it's close to a trail. And so we can, to that extent, because we have a lot of replication both within locations and between sites, we can at least eliminate outliers when we're analyzing the data. And we do that. But there's no way to totally rule out the effects of human beings having access to these plants. The plants are only monitored twice a week during the phenologically active portion of its season. So it's not like people are trampling around every day. We also train people to try to minimize their interactions with the plant. And most, after you're really familiar with an individual plant, meaning shrub number 154, you don't need to get that close to it to monitor it. You can monitor it from a meter away or maybe 2 meters away, depending on the size of the plant and how conspicuous its phenophases are. Yeah, and then I guess I would also hasten to say that should you be worried about this at plants that you would like to monitor at your park, you don't need to monitor a plant twice a week for 6 months in a year. You could choose, for example, to monitor a handful of plants in May, a handful of species and several plants per species, to get a handle on how those species are currently displaying their phenophases in May or in June or in March or whatever localized period of time you want to standardize or select as your monitoring period. And then year after year, you could monitor those species and plants just during that month and examine how the phenology of your park or that location is changing from March to March to March, year after year, so that as you can ask then, okay, as Marches get warmer or drier, how does this affect the number of species that are in flower in March? And so for locations or sites that are particularly sensitive, you don't necessarily need to have a lot of risk of trampling or human impact on an annual basis.
Great, good tips for sure, that we don't need to necessarily, if you're especially at a transition time of year, that's really the important time of observation.
Right.
We have actually a number of similar questions that came together. I'm going to read you each of them kind of together because I suspect you'll be able to kind of answer them all with one answer. One question, Annie was asking, where's the weather data coming from? Is it collected at each site by the observers, or is it just general NOAA data for like the closest weather station? Sarah had asked, what's the source for precipitation data? And again, was that collected there? And Georgia asked about how close was your climate data to your sites, and what sort of resolution do you recommend that you need considering elevational differences and such that might factor in?
Great question. So for the two publications that we have completed so far, we used the PRISM database. That's publicly available from a website created by, I think it's at Oregon State University. And the PRISM data have been used by probably hundreds of publications at this point. And what are available online are for any GPS coordinate in the United States, a modeled suite of climatic variables based on a model that includes the actual data from, I believe, the three closest meteorological stations. The publicly available data are at a grid resolution of 4 kilometers, although they also have a database for sale that is at the 800-meter level, and maybe the National Park Service has access to those data. But for these analyses, we used the monthly conditions for each of our GPS coordinates that were modeled by the PRISM group. And it includes adjustments for the meteorological data based on, I believe, the slope aspect and elevation of the precise site. So it's not like there's a just a single value based on those met stations. I believe there's also an adjustment based on the local topography. The National Phenology Network, just in the last few months, now enables you to not only download the phenological data for any site where it's been contributed, and this is freely available to anyone, but with those data that include now calculations of the first onset date of the focal species that you request data for, the first onset date of the phenophases you're interested in, but also meteorological data from a database that I'm not actually familiar with. It's called Dept, that I assume is NOAA or some publicly available source. But they're all online available climate data that we've been using.
Sure. And just for everyone, just while you were talking, I just Googled PRISM climate data, and sure enough, that very first link that comes up is PRISM Climate Group at Oregon State University. Okay, we had a question about just some of the variables that were chosen. Did you consider using growing degree days or chilling degree days?
So far, we have not done that, and part of the challenge in that is that if you choose growing degree day, you have to choose like growing degree days up to what point. So you could choose, growing degree days is a cumulative value from one date to another date. So January 1st to say March 1st or whatever. So the challenge would be, and this isn't insurmountable, but choosing the date for which you the cutoff point. So for example, for a species that flowers in, say, March, you would want to use the growing degree day accumulation up to, mid-February or something like that. But you'd have to kind of come up with a justification for what day would be your cutoff point. So it kind of achieves the same thing as, say, the mean minimum temperature for January and February being chosen separately. And I don't have, I don't necessarily believe that the variables that we selected are absolutely the best, because as you can imagine, there are an infinite number of ways to select your independent variables. And so we kind of chose ones that were sort of a compromise between what people have done before. Many publications have used mean annual temperature and cumulative annual rainfall as the dependent variables. And we wanted to be more precise than that, and that enabled us to discover some of the mind-bending results that, say, the effects of warm January can be opposite in direction to the effects of a warm February. And using growing degree days wouldn't enable you to detect that, but otherwise I actually do like growing degree days. I just haven't come up with the best way to use them.
Sure. Great. Got a couple more questions here in the last few minutes we've got, and Janice and Forbes actually had a very, very similar question. Again, I'll read them both to you and you can answer them together in one answer. Janice asked, to match a national protocol, is there a minimum sampling frequency? You mentioned 2 times a week. I'd love to start using this with visitors and interp staff, but she's concerned about continuity and commitment required. And then Forbes had asked, does the CPP protocol describe guidelines for survey timing, such as twice a week in March, twice a week in September, for establishing a project? And what about the number of replicates that are necessary for individuals or species at each park?
Okay, so great questions. I just wanted to direct you for the moment where you can find a lot of information on these topics. If you go to the new website, for which this is the URL, where it's about to replace the old one within the next week or so. But in the resources panel, you can find this document called the CPP Phenological Monitoring Protocol, where a lot of these details are discussed. But in a nutshell, the value of monitoring frequently, say twice a week, is that it improves the resolution at which you can define the exact onset date or the transition between when, for example, you do not see open flowers to where you do see open flowers. So let's say you're monitoring on Mondays and Friday, and on Monday you respond to the question, do you see open flowers, as a no, and then on Friday you return to that plant and you do see open flowers. So you go from the transition of a no to a yes. That means that somewhere in that 4-day period was the onset date of flowering, but you don't know exactly what that date was. So you have an onset date, you might call it Friday, but the resolution or the precision, the accuracy of that is potentially off by 4 days in case, in the event that it had opened its flowers on Tuesday. So there's some error in the value that you get. Now, if your primary goal is education, and that's a totally valid and important goal at any national park, you don't need to worry about the frequency. Just monitor when you can, get the data into Nature's Notebook when you can. It's not time sensitive when you enter the data. And even if you monitored a given species once a week or once every 2 weeks, that would give us a snapshot of the phenological status of that plant on that date. And while individually we might not know the onset date of that species, if we have those of that individual, if we have those snapshots for many individuals of that species across the state or the nation, then we can begin to put together a picture of what that whole species is doing at the seasonal level, where it starts to flower first, where later. So don't feel like you're compromising or giving bad data if the frequency with which you can monitor is only once a month. The value of having hundreds of people monitoring means that not everyone needs to be monitoring twice a week. We set up a really high bar for these first few years of monitoring because we wanted to be able to get data that we could analyze scientifically, and I wasn't even sure we'd manage to hit that bar. Yeah, I hope that answers your question. But please take a look at this protocol. Take a look at the report on pilot phase activities, and look at some of these other park reports to see how you could replicate even a portion of what we did in one of our pilot parks at your own location.
And I pasted that link into the chat. So if you expand your chat box there, you'll see the link. You can copy and paste it right into the browser to access any document.
And just to show you just to scroll through a couple of the other points, if you go to Phenology in the Parks and then click on, say, Joshua Tree, I've preloaded it here, you can see the locations of each of the places where we're monitoring, the individual plants and their locations. And then if you select any of the individual species, you can get to our page about that species where you can download the data sheet and the 2-page species profile for that species. And going to the Meet the Species page, you can get to our list of all 30 species. And again, through that location, get to the data sheets and the species profiles. So please explore this dev site, particularly the resources, and I think you'll find a lot that will answer any question you're likely to come up with if you explore it thoroughly. But also, please feel free to email me should you reach an impasse that you want to some help overcoming.
Yes, thank you for that. I know we are running out of time. There were still a couple questions, but I'm glad you mentioned that we can email you if we have follow-ups. I know Sarah was particularly interested in some publicly accessible R code since she's doing some similar data collection on the East Coast. And it'll be okay when the follow-up email to include your email address so they can contact you directly?
Yeah, sure. Yep.
Mm-hmm. All right. Okay, that's great. And then just a caution, I did receive a message for someone else involved with the development of the website They say that with that development link, it looks just like the live site, but if you try to create an account, it's not going to work, and you might not see all the info. So for now, just I followed up with posting the current link, which doesn't maybe, it's not going to look quite like you or have everything there, but it will be updated soon. So don't do much on that dev site. Use the live link.
Right, but for the resources, go to the dev site, and you'll be able to download these websites. You just won't be able to create a new account and password.
Yep, for reference only. Otherwise, use the live one. Thank you. So we are out of time, so I'm going to cut you off there. I got a number of people in the questions box that just said thank you. That was really great and really helpful. People are really excited to hear about this project. So we really appreciate that for speaking with us a little bit today.
Great. Thank you so much, and thank you everybody for your time.
I just want to remind everyone you will get a follow-up email in your inbox that will have a link to download the webinar recording as well as the presentation, and we'll have Susan's email address as well. Next month will be January 14 will be the next of our webinars. The topic is still a little to be determined. It might be about pikas and a pika report coming out later. It might not be. We're still working on that. But January 14. Stay tuned after the new year for an email from me confirming what the next topic will be. So thanks again, Susan. Thanks to everyone for joining us. Wish you all a great winter, happy holidays, and we look forward to seeing you all in January. Thanks so much, Susan.
All right. Thank you, Matt. Bye now.
Bye.